This is an Escherichia coli bacterium. It is commonly found in the human body as part of the intestinal microflora. Its cell contains one molecule of DNA closed in the cycle, as every cell of all prokaryotic organisms. It is because of this molecule that this bacterium has been able to survive on Earth for more than 100 million years. The basic building blocks of DNA are these molecules, nucleotides. They consist of three main segments: a nucleobase, a sugar called deoxyribose, and a phosphate, phosphoric acid residue. The nucleic base is a nitrogen-containing heterocyclic compound. In DNA can be found these following types: purine-derived bases, adenine and guanine, pyrimidine-derived bases, cytosine, and thymine. The specific binding properties of these bases allow the transfer of genetic information in the way it exists. Based on the Watson-Crick base pairing, guanine is always coupled together with cytosine by 3 hydrogen bonds, and adenine with thymine by 2 hydrogen bonds. Now, if we overlap the pairs, we get almost the same shape. This ensures that the created chain will not bend or bulge, as would be the case with any other combination. The bases are linked to the sugar component by an N-glycosidic bond. This molecule is called nucleoside. The nucleotide is then formed by bonding the phosphate with an ester bond. Let's now imagine the nucleotide as a structural formula. Once we number the individual pentose carbons, we can see that the nitrogen base is bound to carbon 1 and the phosphate is bound to carbon number 5. Additional nucleotides are linking to the carbon 3. This end of the thread is called 3-end. On the other side, the chain continues by bonding to the phosphate, which is connected to the rest of the molecule via carbon 5. Therefore, the last unoccupied phosphate in the chain is called 5-end. Nucleotides bind together in the polynucleotide chain by phosphodiester bonds. Both ends of the polynucleotide fiber determine its polarity. The chain transcription and synthesis always proceeds from the 5-end to the 3-end. The second chain is completed during replication according to the base pairing rule. The synthesis proceeds again in the direction from the 5-end to the 3-end. The strands run in the opposite directions to each other. They are anti-parallel. Due to the shape of its bonds, the resultant double strand is forced to twist into a right-handed double helix. This helix has exactly 10 nucleotides per turn. The DNA molecule of Escherichia coli consists of about 4.5 million nucleotides, and its length is 1.6 millimeters. The human genome contains 3.2 billion base pairs with an approximate length of 1 meter. It is contained in almost every cell of the body. If we combined all the DNA molecules in our organism, the resulting chain could travel to the sun and back 300 times. It encodes not only a recipe for all the necessary proteins that make up the individual's body, but also its physical and psychological properties. Deoxyribonucleic acid is the real key to life.